Okay, let's get started with the orchestrating the design, make, test cycle, automating drug discovery at scale. My name is Nila Lê, and I am the Scientific Marketing Manager here at HighRes. Thank you for taking the time to join us today. Before we begin, a few housekeeping items. This session will last approximately 45 minutes to an hour. If you can't join us for the whole time, that's okay. We'll send you the recording as soon as it's ready later today. We'll have a dedicated Q&A section at the end, but feel free to go ahead and populate the Q&A function throughout the presentations today. And the webinar is being recorded, and like I said, will be shared with everyone at the as soon as it's ready by end of day today. All right. So today's webinar is a collaboration between Iambic and HighRes, two companies at the forefront of laboratory automation and integration solutions. We're excited to bring you insights from our combined expertise in helping research teams streamline workflows, enhance precision, and accelerate discovery through connected systems. In today's session, we'll explore how connecting disparate systems and data streams can transform your laboratory operations, real-world applications and results from Iambic's automation journey, technical perspectives on integration and work cell design, practical insights you can apply to your own lab automation challenges. Now, let me introduce our speakers for today. First and foremost, we have Joanna Vo from Iambic, who will be our lead presenter. Joanna is a senior associate scientist at Iambic Therapeutics, working in the sample management team. With nearly 4 years of experience with the company, she has played a key role in building and advancing Iambic's design, make, test, or DMT, platform through instrument implementation and software development. Joanna, would you like to give us a brief overview of Iambic and what you do?
Thank you for that very kind introduction. Hi, everyone. Like Nila mentioned, my name is Joanna. I work at Iambic. So just a bit about Iambic. We are a team of actually not 80 strong anymore. I think we're about like 100 strong with colleagues all around the globe, quite the global company. We have headquarters in San Diego, but also in the UK and Bristol. But like I mentioned, we have colleagues like in Brazil and in Indonesia, just all across the globe. We actually started off as a software company in 2019, but decided we have some pretty strong technology, so why not take that in-house and become a tech bio company? We opened up our labs in late 2021, started our first program in 2022, and in just under 2 years, that program has a asset in phase 1 clinical trials as of 2024, and we're experiencing quite the success as of today. We also have a fleet of other programs under our belt, but regardless of targets, if you can go to the next slide, please. Our mission remains the same. At Iambic, we're dedicated to creating breakthrough technologies to chart new paths to superior medicines and to really disrupt the way drug discovery is done. And I'll hand it back to you.
Thank you, Joanna. That's a great foundation for the folks in the audience today who haven't heard of Iambic before or are not quite familiar with what y'all do. So before we jump into Joanna's presentation, let's briefly share how HighRes fits into this ecosystem. Okay, to do that, we have Corriveau here, who is a lead application specialist at HighRes, where he designs and delivers complex automated work cells that enable high throughput scientific discovery. With nearly a decade of experience integrating robotics, instrumentation, and data systems across diverse scientific domains, Eric bridges the gap between engineering and research to help laboratories scale their science. His work focuses on translating scientific needs into robust automated solutions and advancing orchestration through HighRes's Solario OS platform. Eric, take it away.
Thank you for joining. If you're new to HighRes, welcome. If you're an existing customer, it's great to see you again. I'm here today to introduce HighRes, highlight some challenges we see in modern labs related to orchestration of the DMT cycle, and discuss what tools exist to create more resilient internal processes and reduce overall cycle times. HighRes has been around for more than 20 years, and in that time, lab automation has evolved dramatically and in some ways, not nearly enough. Despite the technological progress, automation still exists in pockets, right? Too few organizations take an automation-first approach, not necessarily implementing large-scale robotics, but actively considering what could and should be automated across every stage of science, from experimental planning to execution and eventual analysis. You know, for a while, we've been known as the premier provider of automation solutions, but our role today extends far beyond building the robotic systems of our past. At our core, though, our mission has remained consistent. We accelerate the rate at which life science organizations impact human health. That hasn't changed. How we deliver on that mission has evolved, though. Today, we're focused on enabling laboratory operational excellence, unifying physical and digital automation to streamline the entire scientific life cycle. And we empower every team member now, not just the automation engineers of the world. Whether it's scientists, operations staff, IT folks, our goal is to help them plan and execute efficient, reproducible science at scale, all through an automation-first mindset that meets science where it happens. This slide captures the three pillars that define the HighRes experience: innovation, accessibility, and reliability. Innovation is about how we combine cutting-edge robotics with intelligent software to fundamentally change how labs operate. Whether it's AI-augmented co-pilots, digital twins, or autonomous robots, we're leading the shift towards the lab of the future. But importantly, orchestration opens up more possibilities than just systems. It's about creating new pathways to automation that include smaller targeted solutions and even process or data-only implementations. Accessibility is key to expanding the impact of automation. Today, fewer than 500 labs globally truly run large-scale automation. Why is that? Well, because historically, these systems have been complex, intimidating, and expensive, and that in itself has been a huge barrier to accessibility and adoption. We've all heard the stories of projects in neighboring labs that didn't deliver, and scientists who are hesitant to risk their own research on something unfamiliar. That's the reality we're changing. With flexible plug-and-play systems, open APIs, and modular orchestration tools, we're making automation approachable for every lab, whether that means a small benchtop process or a fully integrated environment. Our third brand promise is reliability. Scientists are quick to abandon tools that don't work the first or second time. But with over 20 years of experience powering high throughput environments, our systems are proven for uptime, precision, and reproducibility, and we're backed by long-term customer success. Reliability builds trust, and trust drives adoption. Together, these three pillars, innovation, accessibility, reliability, are how we're redefining what automation means, not just building systems, but orchestrating an ecosystem that helps every scientist automate with competence. Now let's talk about some of the problems we see in modern labs and what could be possible with efficient orchestration tools. You know, over our history, HighRes has proven our ability to automate processes on robotic work cells. You want to run an ELISA? Great. Here's a work cell that does that. But this is often only one piece of the DMT cycle. This is the test, and perhaps one of many tests. And unfortunately, this isn't the only piece that R&D labs must manage within the DMT cycle. When will the new molecules be ready for test? Will they be in the correct format? When they arrive, will my system be busy processing samples for another program? If the work cell is already busy, how do I get my data in time for my meeting, my data review meeting on Friday? Is there a validated process using manually loaded benchtop instrumentation? Is there equipment in another lab we can leverage? This is what we're thinking about when we consider the problems associated with planning. Modern orchestration tools should reduce the time it takes to get to execution by exposing entire organizational capabilities and managing the reservation of those associated resources. All right, great. We have a plan. Now it's go time. But what happens when things don't go according to plan? The success of many of HighRes software products relies on features that minimize sample and data loss even when things go wrong, contributing to more resilient processes and shorter cycle times because, frankly, you don't have to redo work. Let's consider one case, the case of a plate washer that breaks down on your robotic work cell. And now I need to divert my samples to that previously mentioned process using manually loaded benchtop instrumentation. What does this manual process look like without an orchestration tool? Well, fingers crossed that the correct settings are used on the instrument, fingers crossed that the correct sample plates are used, and finger crossed that the generated results ultimately make their way to your data storage infrastructure. You spent all this time building robust data pipelines into and out of your work cell, but when anything must be done manually, the entire system falls apart. Well, what if this wasn't the case? What if the only difference between these two processes was a human loading the plates, not a robot, and our automated data pipelines remained intact? To us, this is an important piece of execution that reduces DMT cycles by avoiding delays due to resource limitations and errors due to manual data handling. Let's talk a little bit more about these automated data pipelines, specifically the product of the test phase, the generated results themselves. In many labs today, this is where automation ends. Get your laptop out, open up the results file, create some nice charts in Excel. Perhaps you have a macro that does some of this for you. Perhaps you have a SharePoint driver somewhere that you upload the data to. And this is all before you really start to do real science, right? What is my data telling me? What insights can I gain from it? What should my next experiment be? These are the types of problems we think about related to data analysis. Modern software tools should have robust and proven APIs that reliably expose results data, as well as assay-related metadata to external informatics systems right away, so analysis can happen automatically and in real time. Before we dive into the software, HighRes software offerings, I want to touch on our hardware a little bit. So as I alluded to before, HighRes has a reputation for providing the large-scale enterprise level automation, such as our Nucleus platform that you'll see here on the right. But if we want to make automation truly accessible to every lab, then that cannot continue to be the only thing that we offer. What type of tool would be appropriate for a scientist that's new to automation? You'd want a tool that is designed around your lab, not a tool that your lab is designed around. And you'd want an easy connection of devices that already exist in your lab. You certainly don't want to concern yourself with correctly setting up a robot teach point. That's what we aim to deliver with FlexPod, a platform designed to be parked next to a set of devices that already exist on your bench. Equipped with device-specific instructions and vision-based robot auto-teach capabilities, setup is simplified, making automation accessible to scientists where it previously wasn't. And of course, we have products in between these two extremes, between FlexPod and Nucleus. Iambic chose to start their automation journey in the middle of these two with our Nucleus FlexCart platform. The FlexCart platform itself includes an A-cell collaborative robot on a cart base that can be easily moved around your lab. Shelves surround the robot to hold various pieces of instrumentation required for the application. In Joanna's case, she's preparing assay-ready plates ahead of test. And you'll see her FlexCart is equipped with many of the devices you would expect for this specific application: sealer, peeler, centrifuge, a dispenser for solvent backfill, ambient storage for source and destination plates, and of course, the acoustic dispenser itself. Its small footprint is great for labs where space is a premium, but despite the density of the devices on the platform, user access for either offline use or maintenance activities is made convenient by the use of slides and turntables. Additionally, the FlexCart is designed to grow with you, and in Iambic's case, they chose to later add a second acoustic dispenser and additional storage to enhance productivity and walkaway time. As an alternative, a FlexCart can be docked onto another system and used as a subcomponent of an overall larger workflow or system. Alrighty, let's jump back into software. Iambic is leveraging Slurry Scheduler, the most powerful dynamic scheduler on the market, to plan, execute, and report the creation of assay-ready plates later utilized by their downstream testing cascade. Scheduling software is a tool used to perform these steps on the robotic work cell itself. Processes can be easily designed within the graphical drag-and-drop protocol designer. And once a process is defined, runs can be created to tell the software which plates to execute the process on. After runs are created, they can be simulated to predict system performance and answer questions like, how long will my run take? How long will my source and destinations plates be unsealed and exposed to ambient conditions? The simulation engine collects data on previously completed runs to make later simulations as accurate as possible. Once a run has been started, Solario Scheduler's fleet of error recovery tools is available to you, both manual and automatic tools, to help avoid ruined samples or lost productivity due to device errors. And finally, every modern installation of Solario Scheduler comes standard with the REST API at no additional cost. Joanna system sits at the intersection of make and test, making it critical for the system to have accurate source data coming in and report generated destination data coming out. The API enables automation of data pipelines into and out of the work cell, eliminating errors all too commonly associated with manual data handling between different systems. Creation of assay-ready plates is really only one step in the DMT cycle. So, well, what about the rest of it? While Iambic uses their own tool that they built in-house, which Joanna will tell you more about later, we want to talk a little bit about Solario OS and its relevant modules. Solario OS Workflows is a module that can enable sub-processes, such as the creation of assay-ready plates, to be stitched together into a larger overall process. Sub-processes can be Solario scheduler protocols and runs, like we just talked about. They can also be manual tasks or steps performed on a benchtop device loaded by a lab operator. Wherever possible, Solario OS remotely controls instrumentation and automatically collects results, automating the data pipelines of these otherwise non-integrated steps. Requests can be submitted into the larger workflows, and data flows seamlessly between all the associated sub-processes. After requests are submitted, your work week can be planned using the calendaring features. Solario OS Reserve enables you to manage lab resources such as work cells, benchtop devices, etc., against the demand of workflow requests. And finally, integration with external informatics systems is made even easier with a centralized API. Regardless of where data is generated, either on a Solario OS managed benchtop device or any of your multiple Solario scheduler systems, or at whatever step the data is generated, either in make or test, the data is aggregated and available through a single endpoint with all the metadata surrounding the generation of a given results file. With this approach, you no longer need to build individual connectors between the components of your automation fleet and your external informatics system. If your components are part of a Solario OS ecosystem, then your informatics system already has access to the data. Alrighty, well, that's all I have for today. I hope I've been able to demonstrate to you the new types of problems HighRes is working to address through automation and the hardware and software tools we're building to enable more resilient and efficient DMT cycles that ultimately accelerate the rate of science. Back over to you, Nila.
All right. With that context in place, I'm excited to turn the floor over to Joanna for the main presentation of today's webinar, and probably the reason why you're all here. So as scientific research becomes increasingly data intensive, the need for advanced lab automation has never been more critical. Joanna is going to walk us through how connecting disparate systems and data streams drives real scientific outcomes at Iambic. She'll share insights on how automation can drive efficiency and reduce human error, strategies for providing researchers with deeper insights through connected systems, and real examples from Iambic's automation journey. Joanna, the floor is yours.
So thank you, Nila and Eric, for laying out the context. And like they have alluded to, automation is really more than just the robotics. It's the software as well and how we can connect those data streams. So let me take you through our drug discovery process at Iambic and show you how we're automating the cycle of AI-powered innovation, getting us from insights to drug candidates. All right, so just a bit of background about how drug discovery is traditionally done. I like to think of it as sort of like a linear process. You identified, what sort of target you want to hit, look for the leads, optimize on, different characteristics, it's like better selectivity, optimize on like activity, ADME properties, and whatnot. But at any point in the step, we are bound to hit a hurdle. That's just how science works. And sometimes these setbacks can, make us lose a couple weeks, a couple months, or maybe even set us back to the beginning. But at Iambic, we figured, why go in blind? Could we build something that can help us predict all these properties right from the get-go, right from the design stage, so we can de-risk these molecules before we even make them? And we did just that. Couple that with our high throughput chemistry and biology platforms, along with automated data handling, remixed this linear process into an iterative design, make, test cycle, allowing us to make and test hundreds or thousands of compounds in just 7 to 10 days. And every time we go through this cycle and the data gets uploaded, the models are retrained and get smarter and smarter, enabling us to make better molecules, better for success in the clinic. So a bit about those AI technologies that we have. One of them is Neuroplexer, which is a protein ligand prediction tool that will show us how interaction is crucial to determining efficacy of a molecule at a specific protein pocket. So give us a, one GPU in a couple seconds, and we can visualize what these interactions will look like, which would save us a couple weeks and a lot of money, especially if we're trying to make a compound and sending it to X-ray crystallography for just one snapshot of that. We also have Enchant, which is a multimodal transformer model trained on many different data modalities across multiple contexts like chemical, biological, preclinical, or clinical data, to name a few. So being able to access and interpolate that data right from the beginning of the pipeline gives us an advantage to predict and make a powerful therapeutic and to see how it would go to and through clinical trials. But that's not all there is to the equation. It's just that top part you see here, that little hexagon up there. But we also have, like I mentioned, our high throughput chemistry, biology, and data platform. So what is it that blends this all together and automates the cycle so that it's seamless and that we can iterate on a 7 to 10 day time scale? And the answer at Iambic is what I like to call our secret sauce. It comes in the form of an in-house built web-based application called Insights. And so at Iambic, automation, like I mentioned, goes just beyond the robotics and the AI. It comes as the symphony between the AI, the scientists, the robotics, with the software as the conductor that drives all these components together. Our custom-built software is an extremely powerful tool used by everyone in the company to make all the discrete steps of the DMT cycle happen at Iambic. As you can see on the left here, it is in every part of that cycle in the design stage where we can visualize all those ML models. It also is aware of our in-house and external chemical inventory to make these compound libraries happen. It also partakes in the make stage where we can choose from a menu of various HT-compatible chemistries and can generate robotic instructions for our robotic liquid handlers. We can also use it as a request system where biologists can request compounds that we make to test in cellular, biochemical, and ADME assays. And this is also where we upload our data to retrain our models. So it is very integrated. Like I said, the full integration of the design make test cycle. So I will show you exactly how that happens on Insight. But first, just to lay some context, I'll go over two main workflows that we have here at Iambic. This first one, I won't go into too much detail since it doesn't completely showcase the capabilities of Insight. The first one is nanoscale, also known as direct-to biology, where we're making 384 to 1536 compounds at a time. So a whole bunch, big wham. So for this one, it's typically applied in scenarios where we're exploring like a large chemical space. We're creating about 5 microliters worth of chemical reaction in each well and testing that crude material directly. So a very quick turnaround sort of workflow here. But what I really want to go over is the microscale process. This is a more involved process. We're making up to 96 compounds at a time, about 2 to 10 mg of product for each successful reaction. Now, imagine at this point, we used our AI models to come up with a chemical design, or we found this pocket or chemical space we want to explore. And then we have a recipe, if you will. So in order to make this compound library, we have to start by prepping all the materials. As you can see on the top here, we have all our starting materials that we will tap out into these little tiny 2D barcoded shells that can hold up to a mil at most. Yeah. And so the reagent management team will do this step here. And once all the materials have been prepared, it gets handed off to the high throughput chemistry team, in which they will execute this reaction on a liquid handler, the Opentrons, that's housed in a glove box. So the reason why we do our chemistries in a glove box, it's under a nitrogen environment, because a lot of the chemistries that we do are air and water sensitive. So we want to be able to remove those components as much as possible. Once the library has been executed, we let it cook. We have a lot of food allegories here at Iambic. Once the reaction has cooked and compounds have been made, the library gets handed off to the analytical team, in which they will do pre-QC to determine the best purification methods for the compounds and reactions. They'll do their pooling, the purifications, and dry down the materials so that it'll get handed off to the compound management team to do the distribution process. This process is where we take the purified material, we create 10 millimolar stock for assay testing, and if there's any extra, we put them in an amber vial to dry down for long-term dry storage in the case we need to come back and make more liquid material from it. So as you can see here, there's a lot of handoffs between different teams, and you can imagine if we're making hundreds of compounds a week, this can get very messy really quick. And so how we streamline this at Iambic is we utilize Insight to help with all these handoffs. So everywhere where you see this blue arrow throughout this entire flowchart is where Insight is involved. So let's get into the nitty-gritty details of how Insight plays into that. Okay, so first off, we have our, we're in the make stage right now. So like I mentioned, we have our AI-informed chemical design. So the chemists and machine learning scientists alike can come into our high throughput experimentation electronic notebook, or HTELN for short, where they can set up all the reactions, put in all the materials they require to make said reactions, and they can play around with these rectangle panels here to set up the exact recipe required to make the designs that they want to make. They can set up, reaction temperatures, conditions. They can choose from a wide menu of HDE compatible chemistries like Suzuki, Buchwald Hartwig, and reductive amidation, to name a few, and the ELN will enumerate all these products for them. So once this recipe has been set, the status of the library goes from the planning stage to ready to tap stage. This will notify the reagent management team via Slack notification, as we have Insight integrated with Slack, to tell them that, okay, this library is ready to go ahead and start preparing all the materials. As Insight is aware of all our in-house chemical inventory, it will populate with all the best available source barcodes, as you can see here, and the reagent management scientists will gather everything they need, and they will scan in the source barcode as well as the target barcode, so those little glass shells that I mentioned earlier, and Insight will tell you exactly how much you need to tap into that vial and in which sort of labware. And in some cases, if you need multiple aliquots of the same material, it will tell you that as well. So in this way, Insight has real-time validation and will validate barcodes and the work we do as we go, which is super helpful. Again, we're handling hundreds of compounds every week, so it's easy to mess up. We're only humans, so having Insight do this check for us is very, very helpful. Okay. So once all the materials have been prepared, now we're ready for execution. So the library goes through another status change, ready for chemistry, and this will notify the high throughput chemists that all the materials have been set. They are good to go and execute and make these compounds. So once we have that, the chemist will take a rack scan of all the prepared materials. We'll upload this into the ELN, and at this point, we're still in the same ELN page. So all these different teams are going in to the same ELN interface to do all their discrete steps. And we upload these rack scans into this interface here, and the ELN will automatically generate, oh my gosh, got ahead of myself, sorry. Okay, so the ELN will automatically generate a unique Python script for this library that we can communicate to our OpenTrons' liquid handler here via Python API. So long gone are the days where we're huddled over a laptop trying to code something for each and every library. By having this enabled in the ELN, we're able to execute multiple libraries within a day. And the ELN makes it so that we can easily configure the OpenTrons to whatever conditions we need, because we may be handling different solvents for each different library we do, and we want to make sure that whatever conditions we have set for, like the pipetters, for example, can accommodate solvents of different viscosities. Awesome. And so next, once the chemistry reaction has gone to completion, the crude material then gets handed off to the analytical team in this blue arrow here. And the crude material will now undergo either reverse phase purification or SFC purification, depending on the compounds themselves. Then the purified materials will come back to the compound management team, in which it will undergo the distribution process. In both of these steps, we're still again in the same ELN page, and the ELN will generate TCAN work lists in the form of GWL files or CSV files to execute those steps. So all throughout here, you can imagine the compound themselves are going through different vessels as it makes its way through. It started off in the plate, then it goes into a bar-coded test tube, then it goes into a mini tube and an amber vial. And so what's great about Insight as well is that it can keep track of all of the life cycle of the compounds as it makes its way through. Because you imagine, again, handling hundreds of compounds, things are bound to get lost. But with Insight, we don't have to worry about that at all. We don't have to babysit these compounds. One would go insane if one had to keep track of all of this week in and week out. So with Insight being able to do this, we're actually, at least for compound management, we're able to handle and process many different compounds across many different libraries all at once. We don't care where it came from. We just need to know the batch identifier, and Insight will keep track of all of that as we make our way through the distribution process, getting it ready for the testing stage. Okay, so the last step in our make stage is compound promotion, which some may refer to as the registration of the compound. This is when purified materials go through final QC to make sure it's pure enough to go through the testing stage. Like the previous process, we just get an rack scan of all the compounds we made in those mini tubes, and it'll generate a TCAN file that we just feed into TCAN and TCAN GWL file. And then this plate is handed off to the analytical team. They run the final QC on this and upload the data into the same ELN, and if it passes the criteria coded in the promotion process, the compound then automatically graduates to final compound status and is ready for testing. Okay, great. Now on to the next stage, the testing stage, which quite honestly is my favorite stage because this is where I spend most of my time. So I've gone through the design and the make capabilities of Insight, but the test phase is also another area to highlight through the discovery process. We've built our own request system alongside a LIMS to test the compounds that we've made. Each compound gets deposited in what we call a bucket, which is a logical container that contains its own dilution curve that we've set and built along with a plating schematic. We've built within Insight a curve builder and a plate designer that is flexible and intuitive, allowing us to support a multitude of assays no matter the parameters. As you can see here, this is our curve builder on the left. We put in the assay conditions, we sit down with the biologists, go over the conditions, and we build out a curve. And so we can build a pretty large curve. I think this is something that we actually use for reference control or something. And it's smart enough to know how to make intermediary concentrations, oh my gosh, I'm so sorry, from pre-existing intermediary concentrations. And it can even pull from multiple sources, as you can see here. I know the text might be a little bit small, but it can pull from multiple sources to get to the desired concentration. And the beauty of this is that we just set these parameters once when we sit down with the biologist, press save, and all they have to do in the subsequent lifetime of this bucket is to just put in compounds, and Insight will know exactly what files to make to create the assay-ready plates. Okay, next slide. Okay, so this is just a bit of context for our assay-ready plate workflow, which is not novel and not revolutionary, but I think the way we do it is pretty smart when coupled with our software. So of course, we made those 10 millimolar stock tubes from the libraries. We store them in our Verso, and we pull them out, we thaw them, decap them. Then we put them on the TCAN to create our source plate on an echo plate. So we're plating 10 millimolar stock on a TCAN. That will create our NARP or NARP or near assay ready plate, which we then put onto our flex cart. Okay, this is a very outdated picture. You've seen the better picture before. Before we did have a flex cart with just one echo, but we've upgraded since then. And we use this to create our many assay ready plates. So I'm sure this is not a new workflow for most. But that being said, how are we utilizing this workflow and really maximizing our throughput, as we have done with our automated chemistry synthesis process? We have 2 echoes and 1 flex cart, also not shown here, but shown previously. And with Insight, we're able to do batch processing of requests. So once assay plate requests have been submitted, a compound management scientist like myself can execute on multiple requests at a time. By having buckets defined with their dilution curves and plating designs beforehand, the request system will automatically generate the pick list for the TCAN for source plating and also for the echo for assay dispensing. So this reduces the file handling time and having to jump onto third-party softwares to create a multitude of files. Like if you see me like 3 years ago, I had like 30 files I had to work through, but now I'll show you soon. We just have a neat set of 5 files. So we've also optimized the pick list in such a way that we reduced robot handling time by a third. So dynamic reorganization of the source plate based on the rack scans that we've inputted here to minimize time on the TCAN, and also for the assay pick list in a serpentine sort of dispense pattern, which reduces the number of times the source plate is moved through the HighRes system. And through this as well, we've, by consolidating all these requests together, because often some of these requests have overlapping compounds, we've reduced usage of consumables and precious compound inventory. And Insight does all this calculation for us. Like I really don't have to think. All I have to do is just set it up, and then Insight will do the thinking for me for the rest of the assay's lifetime. As you can see, our system really isn't huge. Like we just added an additional like Ambi store and another echo, and oh my gosh, why did it do that? Sorry. And so, but with such a small system, it's a very mighty system coupled with our software. And with just that, we're able to push out a personal record of 700 assay ready plates in a month. And it's truly a show of a synergistic collaboration between robot automation and software automation. So we're doing lots for a small company with very little instrumentation. Okay, next. All right. So here's just another snapshot of how capable Insight is. So in this use case scenario, we're just using a simple file exchange system. In a previous slide, I was showing you for our chemistry synthesis, that was communication via an API. This is just another kind of application. As you can see on the left here, this is an example of a TCAN pick list that it will generate based on the rack scans that we have. We have a plate visualization right here. And also on this right side here, it's a lot of numbers and text. But you can see here that I've coupled 5 requests together, and it will tell me exactly what sort of plate types I need because that's predefined in the bucket. I need 20 of this plate type, 7 of that plate type, and 2 of another plate type. And all I have to do is just scan in all these barcodes, press save and build, and then it will assign those barcodes to the pick list that I can then input into the instrumentation. So very nifty, very powerful, saves me a lot of time. So just a couple clicks and I can get this nice, neat set of files. But in the past, I've spent like 2 hours trying to make a gajillion files just to get to, I don't know, 5 destination plates. But now I can handle up to, what is this, like 29 plates and even more. Nowadays, even more. All right. If it will listen to me. Okay. So here's just a Gantt chart of those optimized pick lists that I mentioned before. By running things with a serpentine design in our pick list, we've reduced run times by quite a lot. This is quite a large run of 40 plates. We've reduced it from 4.5 hours to 3.5 hours. So big savings, especially again, since we only have the one system with the 2 echoes to run and support all the assays within the company. So I'm running the system down to its bones, like quite seriously, like every day. and overnight as well. So now we get on to the data upload and visualization, because now we've made all these plates. They get sent up to biology team or the analytical team. They undergo their respective protocols, and the data is read. Insight comes back into play here during the data upload and has built-in QC tools as well as curve fitting tools to help scientists quickly upload data for visualization. This saves time on manual analysis, allowing more time for additional assay processing and development. And quicker upload and accessibility to the data also more quickly closes the loop as it retrains our models the moment that data is available and provides more information so that we can enter our next iteration of the design, make, test cycle. So some key takeaways here as we reach the end of my presentation, as I've taken you for a round through our design, make, test cycle. At Iambic, I believe we are at the forefront of AI innovation and drug discovery. We're creating groundbreaking technologies that are revolutionizing the field. We've built and employed multiple AI tools like Neuroplexer and Enchant to orchestrate a more sophisticated approach to the generative design of superior therapeutics. This, in combination with our experimental technologies, creates a comprehensive ecosystem that accelerates and enhances our drug discovery process, allowing us to create better molecules on a quicker timescale. We're pushing boundaries, redefining them, and paving the way for more efficient, accurate, and successful therapeutics that will reach the clinic and hopefully make it through clinic. So thank you so much for listening to my talk. I'll hand it back to Nila for the screen share.
Thanks, Joanna. So now we have reached the point of the webinar where we can engage in some Q&A. Selfishly, I'm curious, Joanna, what was the biggest surprise or challenge that you encountered during your automation and your integration journey?
Yes, this is a great question. So a surprise that I had was how gratifying it is to find all the instrumentation and build out the software to make them all speak to each other. Another big surprise is how much nicer life is when all your automation works as they should. But a challenge to get here, like it's not always a smooth process, is when things don't work out the way you want them to. So sometimes the instrument will break down and we only have an N of one of these instruments. So it can get pretty dicey when something breaks down. But I think what really saves the day is having a great team to work with, not only internally, but also externally. So for example, like this, they're not paying me to say this, but really working with HighRes has been a dream. You guys have been extremely helpful and making sure that all my issues are solved. Like even when I'm not responding to the follow-ups, you guys are like, are you sure you're okay? And you're so dedicated to making sure that everything is working and in order. So that really helps us here at Iambic, especially since we're small and we're starting. We have just a few instruments under our belt. And so yeah.
Oh, that's so kind. Thank you, Joanna. I will thank you on behalf of the HighRes support and service team. Okay, amazing. We have a question from the chat. It looks like a substantial bottleneck here would be how you're working with solid reagent stocks. Is this human operated, reconstituted by TCAN, or actually just solids being manipulated by high throughput hardware? Joanna, would you like to take this one?
Yeah, so this is a great question and a challenge that we are currently navigating right now, especially since we're scaling up in terms of how many compounds we're making in a week and additional, programs that we're maybe planning to put under our belt. So currently, it is a human operated process where we have a very dedicated team members to help tap out all the reagents that we need. We are exploring different avenues like outsourcing this process where we ask another company to tap all these materials for us. It does get a little hard with automation because a lot of the reagents that we do, they're not all beautiful, fluffy powders that can nicely go into a small vial. A lot of them are sticky like oils or like tars, which does make it hard for automation. But yes, this is something that we are trying to streamline, especially, again, like I mentioned, we are trying to scale up our micro scale processes.
Thank you so much. Okay, many questions are coming through for you, Joanna and Eric. The next question that we have is, are your biological and ADME assays also automated?
Yes. So for this, I would say we have instrumentation in-house to try to automate these things as much as possible. Of course, we don't have our like cell seeding on like a high-res platform, but we do have liquid handlers within a fume hood to help like dispense like the cells and whatnot. I think at Iambic, what's really nice is that we always try to look for avenues to automate wherever we can. Some due to the nature of some processes, they do have to be a little bit more manual, but wherever there is a pocket for automation, we try to automate it.
Awesome. We have a few different questions surrounding the software, so both for Insight and Solaria OS. So some of the questions are you currently using Solaria OS, Joanna? And if so, how has this orchestration solution benefited your laboratory's DMT workflows? And then related, can I ask Eric whether the Solario Scheduler API allows access to parameters and data at all levels, from the main Solario orchestration PC to individual instruments? So why don't we start with you, Joanna, answering the question about Solario and Insight, and how you decided to build Insight there, and then we'll go to you, Eric.
Okay, awesome. So we are not integrated with Solario OS actually, but we recently had talks with the team to see how this sort of collaboration could happen. I think one of the missing pieces for us now is, as you can see, with Insight, especially when it comes to assay ready plating, it is a file exchange sort of system. And where I think we could really automate this is since Insight already knows what files we need and we have the protocol set up on our Solario scheduler, the next bit was to have a button that we can just press and it'll just send the orders automatically to Solario scheduler. That way, all we have to do, rather than clicking through the cherry pick wizard on Solario scheduler, if you guys are aware of that, it's quite a lot of clicks. We just have to load the plates up, scan the system, and then the HighRes system will just know to play it. So I think that would be a next step for us. That's something we've tried to build in-house, but we haven't had the time or the bandwidth to as of yet. But we do actually access the Solario API in another way to to access and read the transfer files from the echo post run to make sure that all the transfers have gone through okay. So we use a software called NIME, where we get into the Solario API, get all the run orders that are on the screen, and then run it through and read the files so that we can make sure that all our runs have transferred successfully, or if there's any errors, to create a redo file to recover those failed transfers. So I think that can also be something implemented through Solario OS in the future as well.
Awesome. Eric, would you mind answering the portion about Solario allowing access to parameters and data at the various levels?
Yeah, absolutely. So like I mentioned, every installation of Solario Scheduler comes with the REST API, and part of what you can do through the API is create runs against processes, against protocols that you've built. And when you create those runs, you can establish, parameters with that process and define those parameters as part of the order that you submit into Solario Scheduler. So all that being said, you do have control of the parameters that go into the particular run, and your protocol design defines how those parameters are used within the process. And then, so that's data coming in, and then on data on the backside. So, everything that occurs within an automated run gets uploaded to the event stream. We have an events management system, and all of that data is available to be queried again via the API for particular, event types. For example, you can have operational events that define the set of parameters that were executed on a device on a particular plate, as well as you can have data events where results are generated, and you can go ahead and query the API to give you that results file that was generated. So I hope that answers your questions around parameters and data available through the API.
Awesome, thank you, Eric. Okay, moving on from Insight and Solario OS for now, let's go to a question from James. What tools, hardware/software have you created to validate the quality of significant data you're producing, Joanna, as well as what temperatures do you run your reactions after the Opentron? What device do you use for heating the blocks? So if you can answer that, Joanna.
Yes. Okay. As for the first question, is this in sort of regards to like our biological data output or sort of the more robotic like data outputs that we have? But in the meantime, I will answer the next question. Next question was, sorry, what was it? Oh, what temperatures do we run our reactions at? It varies depending on the chemistry, but for one example, we do run things at like 40 degrees Celsius. And as for what device we use to heat the blocks, it's these, we have these like tumble stirrers that can heat up for our paradox like reaction plates that we just like slip them in so that it can stir and heat overnight.
Okay, and James mentioned that his question related to the hardware and software and the amount of data that you're producing is related to reproducibility.
Okay. So for that, there's a couple of different ways. So we can visualize reproducibility of like all the assays that we are preparing via different QC methods for our instrumentation. So like colorimetric, or even within the data themselves that the biologists will get when they have their data readouts. So oftentimes, if data doesn't look tight, for example, for a particular compound or reference compounds, then we can try to pinpoint, where it is that it could have gone wrong. It could be the integrity of the compound itself, but in some cases, there was a time when we had to essentially recalibrate our echo, and we could see that in the downstream effects, which is not the most ideal case to find out these issues because this is real life data that we're generating that would have to be retired eventually. But yeah, for example.
Nice. All right. I'm noticing time and we have time for one or two more questions. And don't worry, folks, thank you so much for being engaged and for asking so many questions for Joanna and Eric. Any questions that we don't get to today, we will follow up with our team via email, okay? Okay, so the next question is, Joanna, is Insight currently available as a platform commercially via an academic license or otherwise?
Yeah, this is a great question. This is not the first time someone has asked me this. Unfortunately, it is not commercially available. I know it is quite the tempting tool and is very powerful and curated to our needs. But as of right now, it is not.
Darn. Well, you know what they say about roadmaps is that they usually follow where the money is. So maybe in the future.
I'll pitch it to the team.
Yeah, for those who don't know, I believe I have this right, Joanna. Please correct me if I'm wrong, but Iambic started as an AI software company in an incubator, right? And then transformed into like a mini pharmaceutical company.
That's great.
Okay, let's see. I think we have time for one more question, and we'll go to this one for you, Eric. There's been some recent effort towards adopting some universal standards for software to control laboratory equipment like Sila 2. Are any of these software solutions sufficiently developed that you consider them broadly applicable? If you work with any, do you have a favorite among them?
This is a great question, and I'm not surprised that there was a question about Sila and Sila 2 that came out of this webinar. But I mean, to be frank, I mean, I don't know a ton about Sila and Sila 2 specifically for me, but when I think about the ideas behind them, right, I think that they're crucial going forward, right? When we start to think about, like, you know, the lab of the loop, the lab of the future, you know, agentic models, AI models driving processes on automated solutions, you know, standardized ways of creating or of controlling devices is going to be critical for that, right? You know, when we think about these processes today, a huge bottleneck in automating the entire loop with AI is, you know, comes down to control of the individual devices that we're integrating. So I think, you know, I don't think that we're quite there in being able to, you know, implement, you know, unified device control for that type of approach, but they are making good strides towards making that more applicable for various applications. So I hope that answers the question.
Awesome. That's a great question to end with. Alrighty. So I want to thank you again, Joanna and Eric, for sharing your expertise with us today. This was incredibly valuable. I can tell just by the engagement in the chat. Again, here's what happens next, everybody. The recording of today's webinar will be sent out to all of you as soon as it's ready. And the follow-up email will also include a brief survey. You can also scan the QR code here to provide your feedback. I'd greatly appreciate your feedback in helping us improve future webinars. So please spend 2 minutes of your time if you have it. If you'd like to continue the conversation or learn more about how Iambic or HighRes can support your lab automation needs, our contact information will also be included in this follow-up. Thank you all again for joining us today and for your thoughtful questions and your engagement. We hope you found this session valuable and that you can apply these insights that you've learned today to your own lab automation initiatives. Have a great rest of your day, everyone. Take care. Bye-bye.
Bye-bye. Take care.
Thank you so much, everyone.